So thank you everyone for joining us tonight. First of all, I would like to acknowledge the RACI Queensland Polymer Group for enabling us to do these meetings, which is really good. It's quite a great opportunity we have to have scientists from all around the world to share their science with us. So tonight, or this morning in UK, we have Dr. Amanda Pearce presenting us her science. She is a University of Queensland alumni. She also did her PhD there, graduated in 2015 with Professor Chris Turek and Professor Andrew Whitaker. She then moved to UK for a postdoc with Professor Cameron Alexander to Nottingham, and I don't know exactly when, but later on she moved to Birmingham, where in 2019 she was promoted to the position of group leader in Professor Rachel O'Reilly's group at the University of Birmingham. She is focused on bespoke novel synthetic polymers through various synthetic strategies like raft, rope, etc. for in vivo drug delivery. And today she will present tunable and highly functionalizable drug delivery nanoparticles through versatile synthetic routes. Thank you, Amanda. The floor is yours.
All right. Thank you. Thank you for the nice introduction. So, hi everyone. It's a pleasure to be speaking to you today, although this is the first time I've ever done one of these seminars and it's extremely awkward. It feels weird to be talking to yourself in your living room. So, as was introduced, I've been over in the UK for 4 years now, and so what I'm going to talk about today is a combination of some of the work that I was doing my first postdoc at the University of Nottingham, and some of which has continued into my current position at the University of Birmingham, working with Rachel O'Reilly. So before I started, I wanted to just kind of highlight the areas of my research interests that I've developed over this time. So all of the science that I like to do fits around these three themes. So a combination of pharmacy drug delivery using polymer chemistry. So overall, I'm looking at polymer nanoparticles for a variety of different drug delivery applications. And so this started during my PhD at the University of Queensland, working with Andrew Whitaker and Chris Turek, and I was making these hyperbranch polymers with raft chemistry for drug delivery to prostate cancer specifically for my project. And I continued some of this when I moved to the University of Nottingham. So looking at raft still for making these hyperbranch polymers, but also branching out into some ring opening polymerization projects and ways of making different sorts of materials using a slightly different chemistry to make biodegradable materials. And I'll be talking about this a little bit more detail later. Also looking at ways that we can combine these polymerization techniques to make some new and interesting materials. And I'm also interested in different and versatile synthetic routes that we can use to introduce functionality into our materials. And this was an example of some work using some Passerini chemistry to functionalize raft agents. From my time in pharmacy, I've also been involved in some projects a bit more fundamental, self-assembly related, so screening of nanoformulation parameters in order to achieve stable nanoparticles, and also some work in high throughput screening and formulation of difficult to formulate drugs for making sort of solid dispersions and different formulations in that way. But what I'm going to focus about to talk about today is essentially three different projects. So the first was a project entirely based on raft chemistry, and it was my main project that I was employed on at the University of Nottingham. The second project is going to focus more on ring opening polymerization, which was also from the University of Nottingham, and then how these two came together for this combined raft ROP concept and some of the interesting projects that came out as a result of that. So before I started, I just wanted to kind of touch on why we might be interested in combining different polymerization techniques and combining some of these aspects. So raft chemistry or other controlled radical polymerizations are quite well known for they're compatible with a wide range of different monomers, functional group tolerability. We're able to have good control over the synthesis and the architectures that are accessible for the polymers that we're producing. And there's quite a wide size range of materials that we're able to access, but they do have a fundamental disadvantage, especially when we're talking about drug delivery, in that the materials are not inherently biodegradable. Ring opening polymerization, on the other hand, is quite an industrial and pharma-friendly process. It forms polymers which are hydrolytically and enzymatically biodegradable and biocompatible, And these tend to be quite low cost and easy to synthesize polymers, monomers and polymers. And so by combining the best advantages of both of these aspects, we have a way to make biodegradable copolymers where we can still maintain control over molecular weight distribution and the architectures of the materials that we're able to produce. So we can form these kind of novel amphiphilic polyesters with enhanced functionalities and molecular architectures. And so what I'm going to be talking about first is my main project, which was entirely based on raft chemistry. And this was a project within the Biomaterials Discovery Program grant, an EPSRC funded grant at the University of Nottingham at the time, with Cameron Alexander and Morgan Alexander. And the idea of my project was to synthesize a small library of HPMA polymers of a variety of sizes and architectures and take them through a screening process, looking at formulation and characterization of the nanoparticles, some in vitro and in vivo screening to select lead nanoparticles which were behaving the way that we wanted them to in a drug delivery setting, and finally taking these candidate nanoparticles, functionalizing with a drug and assessing them for their efficacy in vivo. So why were we interested in doing this? So we know from the literature that there are a lot of different parameters that dictate the performance of drug delivery polymers in the body. These include factors such as the surface chemistry, the size, the shape or the architecture, ultimately influencing organ distribution, pharmacokinetics, the ability of these particles to enter tumor sites, penetrate through a tumor mass, etc. And however, it's quite difficult to elucidate all of these different aspects and really figure out what it is that's dictating the performance of our particles. So with our project, we wanted to hold one of these variables constant, which was surface chemistry in our case, and synthesize a small set of these different polymers where we were changing the size and the architecture, and we could see which of these effects was having the greatest impact on our drug delivery performance. So we chose to base all of our work on HPMA, which is a monomer known in the literature to be good for drug delivery as an alternative for PEG, and design and synthesize varying polymer architectures where we were keeping the chemistry the same. So we were trying to use a small sets of co-monomers to introduce a different functionality. We explored some non-degradable materials such as this hyperbranched and star over here, quite small, only up to about 12 nanometers, and also some redox responsive materials by introducing these disulfide moieties within our polymers, as these were intended for anti-cancer applications ultimately, and again achieving a size range of up to about 60 nanometers for these particles. So to just explore the synthesis in a little bit more detail, so these were the main four architectures that we were exploring. So the first was a linear polymer. So this was just a linear random amphiphilic copolymer, primarily comprised of HPMA, with this bulk-protected hydrazide methacrylate as a comonomer, and synthesized using a Bitz raft agent which had a disulfide bridge in the middle. So this would give us a cleavage point within the middle of the polymer backbone. Hyperbranched polymers, which we used, again, using the same monomers, the same disulfide moiety as our cross-linker. These could be further chain extended with additional HPMA to form our larger star. And then finally, we also wanted to synthesize a more traditional drug delivery particle, so a core shell micelle. And we synthesized this using a polymerization-induced self-assembly approach. So starting from HPMA to make our macro raft agents, chain extending with the hydrazide methacrylate in situ informed nanoparticles, which we could then also core cross-link using the same disulfide moiety. So we characterized these by a variety of different techniques to confirm that we had not only a wide size range in terms of their hydrodynamic diameter as measured by light scattering. So we could see we had a nice spanning of the size ranges by DLS. Dry state TM also confirms that these were single globular entities, more or less homogeneous in their size. And so then we started the in vitro screening process. So first we were just looking at the stability of these particles in biologically relevant conditions, so in cell culture media. So we measured this using DLS, incubating at temperature in cell culture media over time. And what we could see was that there was no significant changes in our DLS traces over time, indicating that these particles were quite stable to these conditions. To explore this in a little bit more detail, we did some protein binding studies. And this was to give us information in terms of when the polymers are actually in the bloodstream and come into contact with all the blood proteins, etc., that you'll find, and assessing their stability in these conditions. So we used BSA as the model protein, and this was in a concentration range in accordance with cell culture studies. And again, measuring this by DLS, we could see no significant changes in the DLS traces over time, no significant signs of agglomeration. So our particles were quite stable to these conditions. We explored their disassembly, so these were redox responsive. They had disulfide bridges within the core focal points of the architectures, so they were designed to disassemble specifically in a redox environment, such as that within tumor cells. And so we modeled this again using DLS and using glutathione as the reductant. And we could see over time, so this one on the left is representative traces for one of the hyperbranched architectures. We can see a decrease in the size, indicating that our polymers are disassembling into smaller fragments over time. And we could observe this behavior for more or less all the architectures, with the exception of the core cross-linked micelles. And we're exploring that in more detail later on. To start to get a sense of how these materials were behaving as stealth materials, we did some uptake studies using macrophages. And this is to carry on from the protein binding studies. And what we could observe was that there was a time and concentration dependent uptake of our particles into macrophages. For primarily this was dictated by size, but also their surface charge. So the smaller and more negatively charged particles had far less uptake over time than the two larger particles, the linear and the micelle. And however, the highest uptake was observed for the linear particle, which was our closest to neutral particle with a -5 zeta potential. So it seemed that there was a balance of the two factors here when dictating interactions with macrophages. So to further explore this, we then moved into some in vivo studies. And so this was a biodistribution study just in healthy mice. So the polymers were labeled with a Cy5 fluorophore for this study. And polymer solutions were injected into healthy mice, and they were imaged at 1, 4, and 24 hours post-administration to observe whereabouts in the mouse, which organs we would be getting accumulation in. So this is some representative in vivo imaging for the non-degradable architectures, where we can mostly see fluorescence signal coming from the liver, the kidney and the bladders of the mice. And this is expected for quite small hydrophilic flexible materials because they still fit within the renal threshold for clearance, for this clearance route. We also imaged ex vivo at each time point to get some clearer information on the organ accumulation of these materials. And what we could see for these was over time, and as we moved up through the different architectures with their sizes, that we got a change in the organ accumulation and the fluorescence signal that we're observing. So for the smaller particles, most of the fluorescence signal was coming from the kidneys, again, as we were expecting for these small hydrophilic flexible materials. But as we moved up in size range, we can start to see more fluorescence signal coming from the liver, the lungs, and the spleen, so the MPS processing organs. We analysed this slightly more quantitatively or semi-quantitatively by taking regions of interest around each of the different organs of interest. And when we plotted this, we saw something quite interesting. So for the smaller size range of materials, we either didn't see too much accumulation or most of our fluorescence signal coming in the kidneys. And then there was this switch halfway at the halfway points, where from there, the distribution of the fluorescence signals is becoming clearer and increasing into the liver. And so what this told us was that there was an architecture dependence on the clearance route, so either renal or the MPS, and this was in agreement actually with the in vivo data, in the in vitro data with the macrophage uptake studies. So that was quite nice to see. But what we were particularly interested in was that this interface here, where we had two materials, so a hyperbranched and a star material, that were essentially the same size and chemistry, but were differing in their architecture. And this was changing their accumulation in the different organs in this study. So we decided that these two were the materials that we were interested in taking forward into our in vivo drug delivery studies. And this was also dictated by the point that if we looked at the fluorescence in the heart tissue as a sort of proxy for the circulation lifetimes of these materials, that the star, so this material here, had far higher fluorescence signal over time than the hyperbranch, which is the green one here, and this material here. And so what we could see was not only a difference in organ accumulation, but a difference in their circulation lifetimes. And so we wanted to explore this in terms of efficacy in drug delivery. So we took advantage of that Boc-protected hydrazide moiety, which was already within all of our different polymer architectures, removed the Boc group using TFA, and attached Doxorubicin as our model drug. And this was attached to an acid-responsive labile hydrazone bond. which would allow selective release of our doxorubicin in the acidic environment, such as you'd find within a tumour cell. Analysing the dox attachment by NMR and UV-Vis, we could calculate around 10 mol% of doxorubicin attachment. We also characterised these by DLS to confirm that after the attachment of the drug, we still had single nanoparticles in solution that weren't significantly changed from the parent polymer. We did see a slight increase in their physical size. However, this was still within range, and the zeta potentials were still remaining quite close to the parent polymer. We analysed the release of doxorubicin under simulated conditions. So at pH 7.4, so our neutral pH, we didn't observe too much release of our drug, less than 10% for both of them. However, when we increased the pH to 5.5, such as you'd find within the acidic compartments in a cell, we could see this nice controlled release of our doxorubicin over time with no evidence of any burst release. So we then started to explore this in vitro in some cell culture assays. So when we did a cytotoxicity study, so this is using MDA MB231 cells, which is a triple negative breast cancer line, we could see in comparison to the free drugs, so the pink trace, the two polymer drug conjugates had a decrease in the IC50 values after 48 hours of study. And so to further explore this, we also did some fluorescence-based cell uptake studies. And this is some images taken after 2 hours of uptake. And what we could see when we look at the doxorubicin fluorescence signal was we have a significant amount of doxorubicin in the nucleus of the cells. We would expect this because this is the site of action of doxorubicin. So if the dox is being released from the polymers, it will travel to the nucleus. But we could also see some punctate standing of the doxorubicin in the cytoplasm of the cell. And this perfectly overlaid with the Cy5 signal. And so this was telling us that the polymer drugs conjugates were actually able to specifically traffic the drug into the cells, and that we hypothesized this was why we were getting our decrease in the IC50 values over time. So we then looked at this in a slightly more in vivo relevant model using a 3D spheroid model. So if you're not familiar with these, this is where instead of culturing your cells in a monolayer at the bottom of a well plate, you instead culture your cells in these ultra low attachment U-bottom well plates. And what this means is as the cells grow, they start to adhere to each other and they form a tumor-like mass in your well plates. And so this allows you to study in a slightly more realistic setting the ability of the drugs to actually penetrate all the way through the mass of cells so that they can kill the cells all the way through. So we were comparing our free drug versus our polymer drug conjugates. And this is what the spheroids look like under optical microscope. So you can see here at the top our clustered mass of spheroids. So this is at day 0. When we don't apply any treatment or control, they continue to grow uninhibited. When we apply the free doxorubicin, we can see a decrease in the size of our spheroids, and this is mostly from killing of the cells around the periphery. However, interesting to see for our polymer drug conjugates, we actually had complete disassembly of the spheroid structure after our incubation. And this gave us an indication that the polymer drug conjugates were actually able to penetrate all the way through the mass of the spheroid, deliver the drug and kill the cells all the way through. As well as qualitatively, we can also analyze this quantitatively. So using an ImageJ macro, you can calculate the volume of your spheroids from each of these microscope images. And again, you can see this is just for the hyperbranch polymer. With concentration range, we can measure our volume changing over time. And so we can then express this in terms of the % volume change for all of our different samples. And again, quantitatively, we're able to confirm that our two polymer drug conjugates were able to achieve a decrease in the volume or a negative % volume change at a far lower concentration than the free drug was able to. And this was also reflected in decreased IC50 values at the end of the experiment compared to the free drug. And so finally, we wanted to analyze this in vivo. So we established an orthotopic model of triple negative breast cancer. And again, comparing our free drug with our polymer drug conjugate. So before we started, we wanted to confirm that in vivo, the attachment of the drug to our polymer system wouldn't have a significant change on the biodistribution of our materials. So exploiting the Cy5 label that was within our polymers, this was some representative images taken in vivo after 24 hours of administration. And we can see here that we get clear fluorescence signal coming from the kidneys, and we weren't able to observe too much signal coming from the liver, as we might expect if the drug had been causing the particles to become unstable or more likely to aggregate when they were in the body. We were also able to observe that the polymers were able to be taken up into the tumor sites. So this is the tumors on the belly of the mice. So before we started, we've confirmed that the polymers were actually able to reach the tumor site and be retained there. So we then explored two different dosing studies of our free drug, our two polymer drug conjugates, and our vehicle control, which was saline. So the first was a frequent dosing, where the mice were dosed quite frequently over a 2-week period. And the second one was a longer interval dosing, where we spanned out the same number of doses into a 3-week period. What we saw when we were dosing quite frequently was that the free drug and the hyperbranched polymer were more or less the same efficacy, and actually the star polymer conjugate was less. However, when we moved to the longer interval dosing, so we were giving longer days between each of the doses, we were able to achieve far greater efficacy for the polymer drug conjugates compared to the free doxorubicin. And we attributed this to the different circulation lifetimes of the polymer drug conjugates compared to the free drug. It's well known from the literature that you should be able to achieve far longer circulation for the drug conjugates, which gives the drug more of a chance to be able to go to the tumor site and have its action there. So this was the data that we analysed in slight more detail, looking at just non-specific toxicity of these materials as well. So we could observe that they had more efficacy. We wanted to confirm that they also had less toxicity compared to the free drug. So we took the mass of the heart and the spleen as primary indicators of doxorubicin toxicity. This is known from the literature. We didn't see too many changes over time, but there was a slight decrease in the mass of the spleen for the free drug samples, which is known from literature to be an indication of doxorubicin toxicity. However, we could see quite nicely that the mass of the tumours over time was significantly decreased for the two polymer drug conjugates compared to the free drug. And finally, taking mouse body weight as an indication of toxicity. So we could see for the free drug that the mice were putting on less weight and growing less over time compared to the other control groups, which is in accordance from literature. And so just to sort of summarize this body of work, we were looking at a screening process, taking a small set of these different particles, which were all based on this same chemistry, these HPMA building blocks, and taking them a range of physicochemical characterizations, in vitro and in vivo screening, to select two different polymers which were behaving the way that we wanted them to in vivo, and exploring their efficacy in a drug delivery setting. What we were able to see was that within the same chemical built library, that architecture played quite a fundamental role in their drug delivery performance. We were also able to observe reduction of, an increase in stealth behavior and reduction of interactions with blood proteins and macrophages for these PHPMA-based materials, and that our drug-loaded materials had a greater efficacy compared to the free drug in our triple-negative breast cancer models. And so moving on from this, talking about the second project that I wanted to focus on today. So around about the same time at the University of Nottingham when I was doing that first project, there was also another EPSRC project that was ongoing. And this had quite a similar concept. We were looking at different architectures of materials for drug delivery. But in this case, this was a project which was also co-funded by AstraZeneca, a pharmaceutical company here in the UK. And what they were interested in was making materials that were similar to Genexol PM, which is a PEG PLA formulation for the delivery of paclitaxel, approved in the clinic in South Korea. But they wanted a variation which would allow more control over drug incorporation, whether that's from simple encapsulation or attaching a drug. And they also wanted to improve on the synthesis conditions, so removing the toxic metal catalysts, which are typically used, and making these synthesis more accessible for pharmaceutical labs. And so this was a synthetic strategy that project was employing. So taking a series of different PEG initiators, using lactide as the co-monomer as with Genexol, however, introducing this interesting third monomer, which was this cyclic carbonate with a pendant Boc-protected amine group. And by introducing a range of different PEG initiators, we could achieve a range of different polymers. So it had quite a lot of parallels with the first project that I was working on. However, it was using a completely different chemistry, using ROP to make materials which were more biodegradable. And so this is where I joined in. Excuse me. And so the advantage of this cyclic carbonate monomer was that after polymerization and after ring opening, we were left with these pendant amine groups. They could be deprotected using TFA to yield an ammonium group here, which could then be used for a range of different post-polymerization chemistries, such as drug coupling, coupling of other responsive linkers, even forming polyplexes or exploiting the charged groups for the antimicrobial activity, which I'll discuss later. But essentially, we had all these different architectures and materials that we could achieve that had these biodegradable units in there. And so what I'm going to focus on is just two different examples of how we are exploiting the versatility of this interesting carbonate monomer here for forming some simple 1 or 2 step functionalized polymers. So the first was again using doxorubicin as a model drug and attaching doxorubicin to form polymer prodrug conjugates. But in this case, the materials were all inherently biodegradable. So the doxorubicin was coupled to the polymer backbone to form a stable urea linkage. These materials were able to self-assemble by simple nanoprecipitation to form nanoparticles of around 200 nanometers. When they were analyzed in vitro and cell culture assays, again using a breast cancer model and analyzing 2 different cell lines. So MCF7, which is a normal breast cancer cell line, MDA231s, which is our triple negative line, and then MCF10As, which are a normal non-cancerous breast cell line. And what we could see was a decrease in the IC50 values again for our polymer drug conjugate compared to the free doxorubicin. This was particularly apparent for the MCF7 cells. But really nice to see was that for the MCF10As, which are our normal cell, we actually had a decrease in the toxicity. So our polymer drug nanoparticles were able to specifically increase toxicity to cancer cells, but decrease toxicity to normal cells. And we also analyzed these in our spheroid model as before. And what we could see was that compared to the free drug, the polymer drug nanoparticles were able to achieve a slight decrease in the spheroid volume at a lower concentration compared to the free drug. This wasn't as significant as we'd observed in the first project for the really small materials. And we attributed this to a difference in the size. These particles were nearly 200 nanometers, but they still did have a greater efficacy than the free drug. And so to further explore what we could do with this polymer platform and how versatile we could be with this platform, we had a student visiting our lab at the time from the University of Turin. And in the lab that she was working, they were interested in these small molecules here, which were nitric oxide photodonors. And so the idea of these molecules was that in their native state shown here, they were non-toxic. However, under UV irradiation, they were able to release nitric oxide species, which not only were cytotoxic against cancer cells in their own rights, but were also capable of acting synergistically with doxorubicin to enhance doxorubicin activity. And so we thought to see if we could use our polymer platform to specifically deliver this nitric oxide photodonor into cancer cells. And so using the amine group on the end here, we've attached it to the polymer platform using the urea linkages before and shown that these could self-assemble into nanoparticles. But what we did at this point, so remember we also had the same polymer platform, but with the doxorubicin attached, it could form nanoparticles. And we thought at this point, why not exploit the fact that we had the same polymer platform, so they should be quite compatible in terms of co-assembly, and see if we could form nanoparticles or micelles that had both of these molecules we were interested in within the same particle. And so that's exactly what we did, shown here. And so just to confirm the toxicity of these materials, so this is for the nanoparticle here, so the one that only had the NOPD. We could see under dark conditions, so this was just without any irradiation, this was not toxic because we still had our parent NOPD molecule. However, under UV irradiation for roughly 10 minutes, we could get a decrease in cell viability as the nitric oxide species were being released. So attaching this molecule to our polymer platform didn't have any impact on this activity. But the interesting part was when we were analyzing our co-assemblies. So using a variety of different cancer cell lines, we analyzed the cytotoxicity of the free doxorubicin, the doxorubicin particles, and then our NOPD doxorubicin mixed particles under dark and light. And we could see that under light irradiation, we had a significant decrease in the IC50 values for the 3 cancer cell lines that we were assaying. And so this was a really quite a versatile and simple approach for achieving combination drug therapy and synergistic enhancement of activity using this polymer platform. And so we thought at this time that wouldn't it be really interesting if we could combine some of this chemistry that we'd be using here, the ease of the synthesis, the versatility of this carbonate molecule, but introduce that with some of the raft agent chemistry, or the raft chemistry that I was using before. And this might also allow us to solve the problem in that the polymers we were able to make using this synthetic approach were quite limited in their architecture in terms of the PEG initiators that you could purchase. And so we wanted to try and combine these two best aspects, but we weren't entirely sure how. At the time, we also had another polymer, another project that was in collaboration with a group in additive manufacturing over in engineering. And they were interested in this particular synthesis as shown here. They wanted to make a series of polymers which were going to be biodegradable, but they could be photo cross-linked into scaffolds for different tissue engineering applications. And so this was a synthetic strategy that they were proposing. And we looked at this and we thought that this would actually really well suit our need. Obviously, there were these methacrylate groups at the end of these polyesters and polycarbonates. And so we proposed this as our new project to explore. So taking HEMA as initiator as a one-pot and quite simple strategy to introduce groups that were compatible with RACI polymerization into our polyester/polycarbonate materials, keeping lactide as our monomer, and then introducing our Bock-protected cyclic carbonate to form these different macromonomers, which would be amenable to a wide variety of post-polymerization modifications. And so keeping the same concept of this versatile chemistry quite accessible using organocatalyzed ROP, benchtop conditions, initially we were exploring the use of TBD as catalyst, and we were interested in this because it is able to open quite a wide range of different cycles. So we could keep the same synthetic strategy regardless of which monomer we were using. However, it's known from literature that TBD is not only able to catalyze the ROP, but when you're using labile ester initiators such as HEMA, it also catalyzes transesterification processes. So we lose control over our final macromonomer structure. We were able to identify conditions where we could minimize these transesterification side reactions, but this basically involved us keeping the concentration of HEMA in solution to be quite low. So what this meant was that ultimately we were quite restricted in the lengths of the polymers that we were able to synthesize, seeing as we had to keep our initiator concentration so low. It was also known that TBD can remove certain protecting groups, such as the Bock groups, which we had on our cyclic carbonate monomer, so it wasn't going to be entirely compatible with our strategy. So we switched at this point to DBU as our catalyst. It wasn't able to open quite as wide a range of different molecules, but it would work for the lactide and carbonate strategy that we proposed. So proof of concept study, we took a couple of different initiators and just lactide as our monomer in the first instance, explored our conditions using DBU, and we were able to achieve these very short macromonomers with about 5 to 7 units of lactide, with complete control over the monomer structure and no evidence of any other side reactions. And so this was ideal. So moving forward, we then took 4 different initiators as our library to give different properties onto that particular end group of our polymers. So HEMA, PEGMA to give a longer hydrophilic block, and crosslinker, and then also just exploring acrylate versus methacrylate with lactide and our carbonate monomer. And so we can then make a small library of all these different macromonomers, which all varied in their block structure and their block lengths. This would then give us these polymerizable macromonomers, which would be amenable to a variety of different post-polymerization modifications. Primarily, we were interested in these raft chemistries, which would tie in with the synthetic expertise, I guess, we already had in the group. But also these, you could imagine that these could be used for thiolene reactions or mixed Michael additions as different ways of functionalizing these with ease post-polymerization. So as our first proof of concept, we explored just a thiolene click reaction using benzyl mercaptan as our model thiol. We just wanted to see if this end group was going to be reactive, and we could see quantitative conversion of our acrylate end group into the thiolene product. And so this opens up possibilities for as a polymerization technique, for achieving cross-linking of materials, for example, or just as a strategy to functionalize that particular end of the polymer. We also explored the ability of these polymers to undergo RACI polymerization. So first looking at linear polymers copolymerizing with PEGMA in either a random or a block structure. These polymers were able to self-assemble into nanoparticles just using a simple nanoprecipitation. We saw it was quite interesting to see that the block copolymer structure formed smaller and with a narrower dispersity nanoparticles as well. We also investigated if these could be polymerized into hyperbranch structures, as was kind of my interest at the time. So same polymerization monomer setup, but also employing this dimethacrylate crosslinker. And we could see as long as we kept the molar percentage of this quite hydrophobic macromonomer below 30 mol percent, we could still achieve water dispersible hyperbranch polymers. And these were of a size range between about 13 to 30 nanometers. So we kind of successfully set up this synthetic strategy. It was quite simple, quite accessible chemistry, and that we could then use these post-polymerization for achieving quite an unlimited, essentially, array of materials. And so at this point, we thought, you know, what could we do next with this synthetic platform, with these materials? One strategy would be to look at some different mixed formulation strategies. So we now had ways to make polymers of different size ranges and different architectures, but the chemistries were all entirely compatible because they were all built from the same building blocks. So a proof of concept experiment, we explored just the idea of encapsulating the smaller hyperbranch polymers within a traditional polymer micelle by co-precipitating with one of the pegma amphiphilic block copolymers. And we saw by DLS that we were able to actually encapsulate the hyperbranch polymers within there. So this could be quite a simple route to, say, combination drug therapies or delivering polymers which had different release rates. But perhaps more interestingly, and what I'm going to be talking about for the rest of this talk, was how we can further exploit that pendant amine moiety within our polymers for different drug or dye functionalizations, also looking at whether they have antimicrobial activity due to the cationic group, and also how can we further exploit monomer diversity. So there are other monomers that we can introduce into the hyperbranch platform, or other crosslinkers that we can look at to make doubly responsive, stimuli-responsive, completely biodegradable materials, not only for their backbone chemistry, but also for their crosslinkers. Kind of the options were endless at this point. And so I had a master's student that was working in the lab for 6 months, and basically his project was to explore a lot of these different aspects. And so the last project that I'm going to be talking about today is something that just finished up and we just published in Macromolecular Rapid Communications. And this was really exploring the ability for these particles to act as versatile drug delivery carriers, and also exploring their antimicrobial activity. And so for this work, we were interested in taking that same hemomacromonomer structure, using lactide and alcarbonate, but exploring in a little bit more detail how structure chemistry and monomer arrangement would affect the final polymer properties of these. So looking at making hyperbranch polymers as either a copolymer or a homopolymer. So either forming hyperbranch polymers with just 100% of this macromonomer, because we hadn't actually looked at that at this point, due to the fact that they were going to be inherently hydrophobic in their native state. But of course, once we move the Bock protecting groups and this becomes a charged ammonium species, we theorize that this would allow the polymers to disperse in solution. So how those materials would compare as to when we copolymerize them with pegma, a block versus a random structure of our lactide versus our carbonate block. So we had the random copolymer here in this column, or in this column, the block structure. And also redox responsive versus non. So for my first project, I was interested in these disulfide cross-links within the hyperbranch polymer structure. And these are compatible with a wide variety of different biological applications, not only in cancer cells, but also there are different redox potentials in bacterial environments. So we had some materials that were either covalently cross-linked or with this disulfide bridge as a cross-linker. And finally, we wanted to see if we could use this to demonstrate quite a simple synthetic approach to drug functionalized particles by exploiting electrostatic interactions with the ammonium groups after we remove this Bock group here with ionizable drugs. And we used this well-known antimicrobial agent, usinic acid, from the literature as our model drug in this concept. So the master's students synthesized a library of these 8 different polymers, which was exploring all of the different combinations of these aspects. Characterize these by a range of different techniques, just showing some representative traces here. So by size exclusion chromatography, as we normally see for hyperbranch polymers, they aren't perfect traces, but we could see more or less a single population of our polymer species. By dynamic light scattering for the particles that were water soluble in their native state, so the ones that were copolymerized by PEG, we had unimolecular particles of around about 30 to 60 nanometers, depending on the chemistry that we were looking at. So moving forward and seeing how the polymer structure and the polymer properties would affect their performance for this drug delivery. So by removing the block protecting group, as we'd shown previously using TFA, we then exploited the ionizable groups in using acid to try and complex these onto the drugs. And we did this just by mixing the two together in water under sonication for 10 minutes to hopefully form these polymer drug complexes as shown here. And this is what they look like in solution. So usinic acid itself, it's a yellow colored drug, but it's not very water soluble. And so when it's in the vial with water, we basically just see the yellow sinking to the bottom of the vial because it's not soluble. However, when we formed our complexes, we could see a nice dispersion of this yellow color throughout our solution. And this was sort of a qualitative indication that this complexation had worked. We were also able to then analyze these polymers in more detail for their drug content and their encapsulation efficiency, and then their antimicrobial activity in 2 different strains of Staph aureus bacteria, which was a gram-positive cell bacteria strain. And what we could see quite interestingly was for all the different subsets of polymers, depending on whether the macromonomer structure was the random or the block, we had an increase in the drug content and encapsulation efficiency, and then a decrease in the MIC values. So for the block structure, so these are the copolymers with PEG, we can see that the drug content was able to be increased from about 2 to 4 to about 3 to 6 for the block structure. And this had a decrease in the MIC values from about 125 to about 63 micrograms per mil. And for the polymers, which were just 100% of the macromonomer, we had the same trend. So an increase in the drug content and the encapsulation efficiency as we move from the random to the block structure, and a decrease in the MIC from 31 down to about 8 micrograms per mil, which was really nice to see for these. And what we attributed this result to was the charge density of these ammonium groups within the polymer structure. So we can imagine for the random copolymer, these were more dispersed. However, when we had the block with a significant density of these ammonium groups at the end of the polymer chains, we could get multivalent interactions with the many ionizable groups available within the usinic acid. And so we could increase our drug loading and then ultimately their MIC values that way. And then finally, as it's known from the literature that cationic polymers that we want to use for these kinds of antimicrobial activities can also be, unfortunately, toxic against normal cells, which limits their kind of in vivo applications. So we just did an in vitro evaluation in lung cancer cells as a model cell line. And so this study wasn't using the usinic acid complexes. This was just for the charged species. And what we could actually see was that these polymers weren't toxic. When we looked at their metabolic activity after 48 hours, but also their LDH release. And so we would imagine if we had significant destruction of the cell membrane, you get release of LDH in solution and you can detect that. But we weren't actually able to observe these for these materials compared to the Tritnex control. So what we demonstrated here was quite a nice, simple approach for antimicrobial activities that also didn't have any non-specific toxicity. And so with that, I would just like to conclude my talk with, I guess, 3 main takeaway messages. So the first one, we know that controlled radical polymerizations are an excellent strategy for achieving interesting architecture materials with a high amount of control over the synthesis. However, by combining this with complementary polymerization techniques such as ROP, we can introduce greater monomer versatility and functionality into our materials. And then this can lead to extremely interesting materials that have a wide range of different properties and applications. And so finally, I would just like to thank everybody who was involved in this work, both at the University of Nottingham, so Cameron and Morgan, who were my PIs there, Vincenzo, who's been my partner in crime in pretty much every project that I've shown today, and then my new position within the O'Reilly group at the University of Birmingham, Moritz and Stefan, and Jessica and Helen for their help with the antimicrobial project, and you all for your kind attention. Thank you.
I'm the only one who can clap. All the others are. Amanda, it was great.
I can just imagine.
Yeah, they are all clapping. I'm pretty sure it was a great amount of work. How did you even manage to do that in such a short time?
Thank you very much.
So we also have a message in the chat from Linda. What an impressive amount of work, Amanda. I'm not a polymer chemist, so excuse if it's a trivial one. What is the reason for the size change on slide 17? when you formulate the MPs combining both drugs, i.e. from 70 nanometers to 110 nanometers. Also for
Hi,
all the
Linda.
other, if I just may, for all the other attendees, if you just want to ask a question, you can either write it in the chat or raise your hands, and then I can basically unmute you, I believe, and you can ask it yourself. Yeah, sorry, Amanda.
Great. No, that's right. Thanks for the question, Linda. So we thought the same thing as well when we were doing this project, and we think it's actually just due to the size and the bulky nature of the different drugs. So the NOPD moiety here is a lot smaller than the doxorubicin. So we thought when we mix them in with 50% of just unfunctionalized polymers, you don't see too much of a difference. But when you then try to formulate a nanoparticle which has more of the two bulky molecules, the big doxorubicin there, you just get just a physical increase in size because they can't pack together as efficiently to make the smaller nanoparticles. That's our theory.
Thank you.
All right. I also have a question myself, actually. So somewhere, I think in the beginning, slide 11. So when you were functionalizing your hyperbranch polymers with doxorubicin, and then you got something like 10% functionalization, was it that you were targeting for 10%, or was the reaction just such a low efficiency one?
So a little bit of both. So we only had about 20% more for the hyperbranch of the hydrazide group, and about 10% more of the hydrazide group for the star, 10 to 15. So we got about 75% yield through that coupling reaction. And I think the reason we didn't get completely 100% is just the challenges of getting doxorubicin, which is quite hydrophobic, to get all the way into the polymer structure if there's hydrazide groups more internally and in the cross links where it's more dense. So I mean, ideally we would have liked 100%, but it was about a 75% yield was the actual result.
And I guess you had the hydrazide in an excess rather than ending up with free dox floating around, or was it 1-to-1 when you reacted them?
I think we did, it was about a 1.2 excess of the doxorubicin, so we were trying to push it to full conversion, to higher conversion, but we didn't want to risk putting too much free dox in the solution and then having difficulties removing it as well. So we figured a small amount of the free hydrazide groups, especially if it was just because they're in the core of the particles, wouldn't significantly impact on the rest of the study. So.
Sure. And when you did your spheroid studies in 3D, what did you embed the spheroids in? What did you use as a matrix, or were they just free floating in a media, or?
Yeah, so they actually just free float in the media. So for these ones, you need to culture them inside a sort of basement membrane extract. So they form supported spheroids. But after that, yeah, they're literally just floating in cell culture media inside the whirl plate, so they can kind of grow unencumbered, which is why you can get these kind of funky shapes when they grow really uncontrolled, because there's nothing actually keeping them into a round mass.
Sure. So we have another message from Hui Peng. She's acknowledging you for a great talk without any question associated.
Thank you, Hui.
Any other questions from the audience? I'm sure Idris might have one, right? As the most senior person around. No. So maybe just a simple question. Well, we have one, but I will ask. So from those 3 systems that you presented, which one did you like yourself the best?
The 3 sort of overall different ones.
Yeah.
I quite like the last one, and I don't know if it's just because it's the one you're doing now and you always get excited about the work that you're doing at the moment and it's really working. Yeah. But I think it was quite nice playing around with some different chemistries and just looking at the ways that we can change polymer properties just by, you know, working with some different people, working with some different groups, and having the biodegradability and this really good functionality in the end, I find quite interesting. So for me, I'm looking at trying to investigate a few more projects using this particular system. Next time I get another student, planning on looking at this in more detail. I just saw there was a question from Al.
Yep.
So are the hyperbranch polymers biocidal without the attachment of using acids? Thanks for the question, Al. So we did analyze the polymers themselves to see if they were active. We found surprisingly they weren't actually that toxic against bacteria when the drug wasn't attached, and we're not entirely sure why. And I guess maybe that also explains why they weren't so toxic against normal cells as well. Because yeah, we would have expected that they would have actually been toxic as well through those charge groups. And I don't know if it was because we didn't get 100% deprotection of the Boc groups, because we had to keep the TFA step quite small because we didn't want to risk also destroying our polyester polycarbonate backbone. And so maybe we just didn't have enough of those charge groups. And so you really needed the usinic acid there as well. But this is something that we could definitely look into more. But, yeah.
Cool. Do we have any other questions? Anyone? Oh, maybe they still need some time. Maybe I have one more. So somewhere I think around. So when you started to introduce the second system, the project with AstraZeneca, and then you had those branch StarPECs or whatever I would call them. So
yeah,
I know that
kind of
it's not really
things,
yeah, not really the target of this project, but has anyone kind of tried to, you know, cross-link them, make some kind of hydrogels and so on of these?
No, actually, it's quite interesting. I mean, they're already quite interesting molecules because the hydrophilic part is in the core of them. So when they self-assemble, they kind of form these flower micelles. So I guess the next step, so you could actually make these really interesting kind of core cross-linked materials where you've got these loopy pegs on the outside or something. But no, we haven't actually looked at them in their hydrogel for their hydrogel properties because we don't really do that in our lab, but could definitely be interesting. Because I guess this is a pretty common, these kind of molecules are pretty common precursors for making different hydrogels. And the group that I'm working in now, they have some tissue engineering projects where they're making these scaffolds using that. So could be something to look into, to explore, definitely.
Cool. Well, I guess we are not getting anything else. Well, it was a great pleasure having you, Amanda. Thanks for taking the time. Thanks.
Thank you so much. It was a pleasure.
Thanks also to everyone who made their time to attend Amanda's lecture. There will be also a recording of the lecture, so if anyone is interested to revisit it again, just let us know and I can send it to you. Thank you, Amanda. Have a good day.
Great. Thank you. Thank you everyone for your attention. Hope you all have a great evening.